Hello, everyone. Good evening. Welcome to our Cosmic Thursdays. My name is Nandu, and I am in charge of education and public outreach in the Department of Physics and Astronomy at UC Riverside. Some of you might already know me. If you don't, that's completely fine. I am the main organizer of this event. We are super grateful tonight that you are here with us at this virtual lecture. We see that there are many of you who have already been going through some of the virtual events that we have hosted before. Welcome back. And if this is your first time checking us out, we hope you like the lecture and enjoy your time. So this is my pleasure to introduce our speaker today, Professor Edward Schwieterman, who is an assistant professor in the Department of Earth and Planetary Sciences at UCR. Professor Schwieterman got his PhD from the University of Washington with dual titles in astronomy and astrobiology. After that, he worked as a NASA postdoctoral program fellow at UCR and later as a postdoc fellow at Georgia Institute of Technology. After that, he joined UCR as a faculty member in 2020. Professor Schwieterman's research interests include modeling the atmosphere of terrestrial planets, characterizing exoplanets, and also studying planetary habitability. So in this lecture, he will actually be talking about how we can determine the presence or absence of life beyond the solar system, and how we can actually use Earth as a natural laboratory to provide insights into extraterrestrial life. In addition, I would also like to thank our volunteers today that you can also see on the screen. So we have 5 UCR graduate students joining us in the live chat today. They are graduate students working in research fields that are closely related to the topics that we're going to cover today. And they will actually be earning their PhD very soon. So now I will let them to quickly introduce themselves. So first we have Daria.
Hi, my name is Daria Pidorodetska. I'm about to be a second-year PhD student at UC Riverside, and I study exoplanets, habitability, and the detection of biosignature on other planets.
Cool, thank you. And next we have Michaela.
Hi, everyone. My name is Michaela Leung. I'm also a second-year student at UCR. I work with Professor Schwieterman on exoplanets, habitability, and biosignatures. I'm looking forward to answering all of your questions, and that was just my cat, Gus.
Thanks, Michaela. Next, we have Chris.
Hi, everyone. My name is Christopher Jones. I'm also a PhD student in Earth and Planetary Sciences. I'm studying geochemistry, and I am interested in how the Earth has evolved through time and how that can inform our thinking about astrobiology and exoplanet habitability.
Very cool. Thanks, Chris. Next, we have Ian.
Hello, everyone. My name is Ian. I am a graduate student in the Department of Physics and Astronomy. I do observational astronomy. Specifically, I study the environments of massive galaxies in the early universe.
Great. Thanks, Ian. And last, we have Nicole.
Hi, everyone. My name is Nicole Gangoli. I'm a 4th-year graduate student in the Department of Astronomy, and I study very early universe stuff and galaxy formation and evolution.
Great. Thank you. All very exciting topics. So as you can see on the right-hand side on your screen, there's actually a live chat on YouTube that we are going to answer all the questions in there with the help of all of our volunteers. We welcome all kinds of questions in there and also encourage you to engage in discussions. Since we have a relatively large group today, so we kindly ask you to be respectful to others in the chat. The volunteers will be answering most of the questions and pass the remainder, well, some of the questions to the speaker as we go along. So all the questions that are unanswered will be saved for the speaker to answer at the end of this lecture. So without further ado, take it away, Addie.
Yeah, thank you very much, Janan, and all of the volunteers also. I am so privileged and glad to speak to you today about exoplanet biosignatures, the search for life beyond the solar system. And in large part, I am motivated by this question, which I know you're all interested in as well, because you're here listening to the lecture. And the question is, are we alone? And there are different ways of thinking about this. Are we alone in the sense of being intelligent, an intelligent civilization in the universe? Is our planet alone in supporting life, whether that be simple life or complex life? Is Earth alone as an inhabited world with a biosphere? Both really important questions. And I find this quote by our Buckminster Fuller very, very insightful. It says, sometimes I think we're alone and sometimes I think we're not. In either case, the thought is staggering. And as a scientist, we always have our mind open, whether our hypothesis is correct or not. And I certainly hope we're not alone in the universe. And I want to find out. But I'm also open to the idea that perhaps we are alone. And in that case, it's still a really, really important conclusion. But I don't think so. And I want to convince you during this talk that the best way to look for alien life is actually not to look for intelligent aliens, but to look for microbes and pond scum. ET is a microbe. And we need to sniff the atmospheres of alien planets for the signs of those microbes. And so first, to explain why, I'm going to sort of go through our place in space and then our place in time. I say we're looking for signs of life outside our solar system. Our solar system is the collection of planets that orbit our sun. And that our planet is in the solar system. The solar system is one of 100 billion solar systems about in the Milky Way galaxy. That galaxy itself is part of a local group of galaxies. And that local group of galaxies is part of a cluster, supercluster of galaxies. So there are about 10 to 100 billion stars in our galaxy. There are about a trillion galaxies in the observable universe. And that means there are about 10 to the 23 stars in the observable universe. So that's the universe that we can see, whether it be the nearby star or the most distant galaxy. And so that's a 1 with 23 zeros after it. So it's kind of an immense, spectacular number. But even the closest galaxy is far, far away. And so when we're talking about looking for exoplanet biosignatures, we're thinking about the solar systems that are near our solar system. So the hundreds or so stellar systems that are closest to our solar system. So that is our place in space. What is our place in time? I really find this concept called the cosmic calendar very instructive. And the whole point of this, I know this is an overwhelming graphic, but the point of this is think about the entire history of the universe from the Big Bang to today. And imagine compressing that history into 1 year or 12 months. So if we were to do that, if we were to compress the entire history of the universe into a year, our planet doesn't even come into the equation until September of that year. Okay, so almost 2/3 of the way through the history of the universe. And the origin of life followed somewhat shortly after that. But if we think about what life existed, it was simple life almost to the middle of December of this one cosmic year. Okay, so the middle of December is when we start getting multicellular animals. Before that, we have microbes, we have algae or cyanobacteria that may perhaps made oxygen that influenced our atmosphere. And only in the last day did we have the great apes. Only in the last minutes did we have our species, Homo sapiens, last 8 minutes. And the human migrations out of Africa into the rest of the world. only in the last 30 seconds in this cosmic year did we have civilization, meaning agriculture and then writing. And only in the last, only about a second ago did the Europeans arrive in America. One human lifetime in this cosmic year is 0.23 seconds, 0.23 cosmic seconds. So our place in time is a very, very, very small slice, and there was a lot of history before us. If we look at the history of the Earth, most of it was dominated by simple life. And I'll talk about why that's so important and why it influences our search for life elsewhere in the universe. So I conceptualize, often it's instructive to use this equation to think about life elsewhere. And so often we think about, well, what about those intelligent aliens that think like us and communicate like us, and maybe we could pick up their radio beacons, or maybe we could see their spaceships. And so one way of figuring out how many of those civilizations there are was put forward by a scientist named Frank Drake. And he created this thing called the Drake equation. And the idea is that you can multiply a bunch of factors together and understand how many civilizations there are that could communicate via radio in the galaxy. And there are many components to this equation. So the rate of the star formation in the galaxy, the fraction of stars that have planets, the fraction of planets per solar system where there's an environment suitable for life, the fraction of suitable planets on which life actually appears, that's where our biosignatures are, then the fraction where you can get intelligent life, and then the fraction of those intelligent life forms that develop ways of communicating through interstellar means, and then the lifetime of those civilizations. And understanding all of those factors gets you to this N number, which is the number of civilizations in the galaxy. And we can see that the number gets smaller as we go forward. We know for the last several decades about what the star formation rate in the galaxy was. It's about one star per year in a galaxy of 100 billion stars. What about the fraction of those stars with planetary systems? Only in the last 10 to 20 years have we really gotten a handle on this number. And we have done so through a variety of methods, including ground-based radial velocity method. This is looking at the wobble of stars caused by the gravitational influence of a planet. The transit method, which looks for the little miniature eclipses when a planet passes in front of the star and dips the brightness of that star. And those two methods, by and large, you can look at this cumulative diagram, have consisted of the bulk of our discoveries of exoplanets. Not all of these planets, of course, are like our planet Earth. Most of them are, because of detection biases, larger, like our gas giants like Jupiter, or ice giants like Neptune. But many of them are rocky and terrestrial, like Venus and Mercury and Earth. And a subset of those terrestrial planets are actually habitable, or potentially habitable, I should say. And that means that they receive an amount of radiation that's conducive to surface liquid water on their surfaces. So there's about one planet per star. We know this from the Kepler mission and from all of these planet detection techniques that I mentioned. What about the number of planets per solar system with an environment suitable for life? What does it mean to be a habitable planet? So, we have a bias from our own experience of being on the Earth, and we know what makes the Earth habitable for us and for the life that exists here. And that's sort of what we have to use to extrapolate elsewhere. And so the current thinking is a habitable planet has a rocky composition like Earth. It's the right distance from the star, not too hot that the oceans evaporate, so too close to the star, not too cold so that the oceans freeze. It has a thick atmosphere, a greenhouse gas to support warming that atmosphere. In our case, it's CO2 and water. Volcanism to replenish those gases, Potentially other things like plate tectonics, although we don't know how important those things are. Perhaps you can have a habitable planet without, for example, plate tectonics, or continents. And so often you see this phrase invoked, the habitable zone. And so the habitable zone is just this range of distances from a star where a planet with the right atmospheric composition, and that's a key, the right atmospheric composition, could maintain surface liquid water. So if you don't have an atmosphere in the habitable zone, you're still not habitable. The moon, for example, orbits the Earth. It's in the habitable zone, just like the Earth is. Doesn't have an atmosphere, uninhabitable surface. So being in the habitable zone, not a guarantee that you're habitable. And it describes a surface, conducive for perhaps simple life that requires liquid water. Not a lot of other requirements are heaped onto that. So something that's habitable for a microbe is not necessarily habitable for you. So if you had the choice of going to a random planet in the habitable zone, you'd want to rethink that, make sure that that planet is actually suitable for you before you go. And importantly, the habitable zone changes depending on the star. Some stars are brighter and larger and bluer, and the habitable zone is more far away from those. And so the planets in the habitable zone are farther away than, for example, the Earth is from the sun. Some are like the sun, in which case the habitable zone is roughly the same location, as in our solar system. And some are much cooler. In fact, most stars, and I'll mention this later, are redder, dimmer, cooler stars called M-dwarfs. And the habitable zone is actually much, much closer because of the dimness of those stars. So planets have to be much closer in order to get the right amount of radiation to maintain that surface liquid water. And so now we know about a couple dozen, about 24, 25 of these planets that lie within that right amount of radiation, that receive the right amount of radiation, that we could say perhaps with the right atmospheric composition, those planets could be habitable. And this is a nice diagram put together by the Planetary Habitability Lab that shows those planets, their relative sizes compared to Earth and the planets of the solar system, Mars, Neptune, Jupiter, and where they are. Some of these names you may be familiar with. Proxima Centauri b. Proxima Centauri is the nearest star. And so Proxima Centauri b is the nearest possible potentially habitable planet. That's on here. The TRAPPIST, that's Proxima Centauri b. The TRAPPIST planets, you heard the TRAPPIST is a very famous system. It has several planets in this so-called habitable zone. Doesn't guarantee they're habitable, but they're very, very interesting. And we want to take a look at their atmospheres to look for signs of life. So if we think about this Drake equation, we've gotten through kind of half of these terms. We know the star formation rate, we know the fraction of planets per star, and we actually know about the fraction of planets in the so-called habitable zone. Again, it doesn't mean that they're necessarily habitable, but we know a lot more than we did before. And we think about a quarter with huge error bars, but about a fourth of those stars out there have planets in this habitable zone. Okay, so let's go back to this question, are we alone? So I mentioned before, we want to look for biosignatures. So a biosignature, again, is something like pond scum. You could think of it maybe as an unintelligent creature. A technosignature are these aliens, little green men. Okay, we'll talk about that maybe at the end. So again, a biosignature could be something like oxygen in our atmosphere. We'll get into that. It's made by plants, and it influences our atmospheric composition. A technosignature, a sign of an intelligent life, would be something like a radio signal or a laser pulse or a large construct, something like a Dyson sphere, where civilization has built a structure around a star and is collecting all the energy, or UFO landing on your front porch. That would be a technosignature. But importantly, and as I mentioned before, the cosmic calendar tells us that the most likely ET is a microbe. We should be building our telescopes to find pond scum. And so I'm on team pond scum. Pond scum is at least more popular, consistently in public polling, more popular than Congress. So I'm on team pond scum. And so that's where we get to biosignatures. So biosignature, you can think of it as this term in the Drake equation. If we knew the fraction of habitable planets where life actually appears, and we actually are on the precipice of being able to do that. So how can we do that? How can we identify habitable and inhabited planets? This is an image from the Cassini spacecraft of Earth from Saturn. And Saturn is about 1.5 10 AU from the sun, 1.5 billion kilometers away from the sun. On average, it's a little bit further away from the Earth at any given time. It changes, but it's about that distance. The nearest star is 4.2 light years away. That nearest star, of course, Proxima Centauri, has the nearest known exoplanet. That's 40 trillion kilometers away. That's a lot. And so you think just from a billion kilometers away, just from billions of kilometers away, that's the Earth. It's a single pixel, pretty much, pale blue dot. And now we're going to go 40 trillion kilometers away. So how can we take this Earth? This is a model of the Earth. We're condensing all that information, all the continents and the clouds and the oceans down into a single point of light. It's really important. So we'll talk about that. So I think about this a lot in my job. In exoplanet biosignature science, how can we recognize the signs of an inhabited planet? So we can ask questions like, what does life produce? What are the signatures we're looking for? Can a dead planet fool us? Are there abiotic processes, planetary processes that would fool us into thinking that there's life there? And of course, we can't travel, and we will not be able to travel in the near future to any of these exoplanets. How do we interpret these data as we're pixelating that planet with all the information, rich information that you might get if you were there, into a single point of light? How do we quantify those uncertainties? So one thing I like to point out is that biology has fundamentally changed our atmosphere. So I mentioned oxygenic photosynthesis. So there are bacteria and plants and algae that make oxygen in our atmosphere. On Earth, at least, without those things, the oxygen in our atmosphere will quickly disappear. And so we think of oxygen as a sign that life is there. There are other gases in our atmosphere that were only there because of life. And that includes methane from microbes. We think of methane from cows, but it's really from the archaeal microbes that are living in the guts of the cow that make that methane, and they can live other places too. Nitrous oxide, laughing gas, is actually a product of bacteria also. And so this plot here shows you Earth with life and all the gases that life makes, and Earth without life. And look at those huge differences in the amount of gases produced with and without life. So that is not enough alone to indicate life. You have to have some requirements. So yes, you need to be produced by life. We also need to be able to see this thing over interstellar distances. So those trillions, tens of trillions at the minimum kilometers away, probably many more factors of that to get to the stars that we want to look at. Probably globally, this biosignature has to exist everywhere, like oxygen exists everywhere in our atmosphere. Produce observable features, that just means it has to have signatures that a telescope can see. Build up to detectable amounts means that it can't be quickly destroyed by sunlight, broken apart by light from the sun. Inseparable from abiotic processes. That means we have to tell the difference between biology and geology. So if you think about a biosignature application form, here's one for methane, which we know is produced by life. Yes. Can we measure it from a distance with telescopic instruments? Of course. But methane, have you ever been mistaken for a geologic process? Ooh, yes, you have. Methane exists abiotically in the atmospheres of gas giants, and it's also produced in small quantities on Earth abiotically. So we can't just rely on methane. We have to have maybe a combination of gases. And so I think of grouping biosignatures into these categories. So I mentioned gaseous biosignatures. An example is oxygenic photosynthesis. The plants take energy from the sun and carbon dioxide from the air and water, and they make oxygen in addition to their organic matter, in addition to their structures and their cells. And oxygen is processed in our atmosphere into ozone. That's great because we have the ozone layer. We don't get sunburn. That also means something that we can look for with telescopes. There are a couple other categories of biosignatures. So one is something called surface biosignatures. Surface biosignatures are things like the vegetation red edge. And I'll talk about that in a second, but it's just the idea that leaves and vegetation reflect light in a particular way. And that particular manner of reflection is something that we can see with telescopes. Another thing that we might look for is the seasonal changes in those gases. For example, the change in the CO2 in our atmosphere as plants grow in the summer and the spring and the summer and die back in the fall and the winter. And so here are some more examples of these three categories. So atmospheric gases, we mentioned oxygen, ozone, methane, nitrous oxide, which you might be familiar from the dentist. It makes you a little bit loopy, but it's also made by bacteria, and it also produces these observable features in our atmosphere. I mentioned that. Vegetation red edge, other pigments perhaps, and the seasonal changes in those two things. So the difference in leaf color as a function of season is another thing. So here's an example of the vegetation red edge. So the vegetation red edge is a result of the fact that in light that you can't see, leaves are very, very reflective. So the red wavelengths that are just redder than human vision, that's called infrared wavelengths. It's really bright. The leaves are really bright. And these are infrared photographs of vegetation, and you can see just how bright they are, reflecting a lot of light. Earth observing satellites use this fact to map vegetation on Earth as a function of season, and they map things like the plant health and the impact of fires, for example, or of different things of climate change too. And so in the visible, the plants are very, very absorptive. They're absorbing that energy from the sun to make oxygen and to support their photosynthesis. And then they're reflecting very, very greatly in this infrared region here. Vegetation and chlorophyll isn't the only type of pigment that we might look for. There are other things called rhodopsins, which are proteins similar to the proteins that are actually in your eye that allow you to see in color. They actually are in many, many different microbes that use light for energy. One of them is called halophiles, and they live in these high salt ponds. And you can see through the combination of carotenoid pigments, which is a type of pigment, and rhodopsins, you have this deep purple color, or you can have these orange colors or pink colors. And then there are things called, in addition to chlorophylls, called bacterial chlorophylls. And there are photosynthesizers that don't make oxygen, and they use these bacterial chlorophylls. I actually grew these up in little chambers that have no oxygen in them. They're anoxygenic photosynthesizers, and they have these really, really interesting colors. And so you can imagine perhaps a planet that had never evolved oxygenic photosynthesis. Instead, you could have these other microbes with different pigments, and those would be your biosignature, your surface biosignature. And then these are time varying biosignatures, so temporal ones. And so on the right here is the CO2 as a function of your latitude on the Earth and the season. Really what's important is that our planet, if you track the amount of CO2 versus time, you can see it breathing each year as plants grow in the spring and summer and die back in the fall and winter. The vegetation red edge signature changes as a function of season as well, because of seasonal impacts of temperature on leaves. They become, the leaves die back, and in the winter and grow back in the spring and summer. And so how can we see all these things remotely? How do we know what things are made of? So light consists, you probably know this, light consists of many different colors. And we can take instruments like a prism, or in astronomy, we use something called a diffraction grating, which is really kind of like a prism, except a little bit more advanced. And we can break light up into its component wavelengths and look at those component colors in order to determine what's going on in the distant object we're looking at. And so you may have had the experience in chemistry class in school of burning different metals and looking at their different colors. And if you ran those through a prism, you'd see that some wavelengths light up and some do not. And it's the same thing for any molecule out there, including, for example, oxygen, absorbs certain wavelengths and leaves other wavelengths alone. And that unique pattern that's imprinted on the light tells us the composition of the atmosphere. And so if we're thinking about an exoplanet, we can do the same thing. We can take the light from the planet and we can split it into its component wavelengths. And we can look at the wavelengths that are missing and the wavelengths that are there. And we can reference, all the experiments that everyone has done in the laboratory here on Earth, and we can figure out what that atmosphere is made out of. And so I want to show you a spectrum, again, putting the light from the Earth through a prism or a diffraction grating and figuring out what it's made of for the Earth. So this is the reflected light spectrum of Earth. And what's really important here is just, you see these lines and you see these squiggles. So the labels show you what's there. The labels show you these are wavelengths that go from the ultraviolet, so too violet for us to see, to infrared, too red for us to see. This little color bar is basically the amount, the vision that we have as humans. And of course there's a much bigger range of colors and wavelengths. And you can see here the features, imparted on Earth's planetary spectrum, on the reflected light spectrum of Earth by different molecules. Some of them are biosignature molecules. So ozone creates a big signature in the ultraviolet. That's actually really good because that's why we have an ozone layer. Ozone blocks up all the UV radiation, absorbs it, doesn't reach the surface of the Earth. But if you were looking as an external observer, you would also see all that light missing from the reflected light spectrum of the planet, and you would be able to infer that ozone is there on that planet. In the same way, you can look at these little signatures for oxygen and water and carbon dioxide and methane and nitrous oxide. And through this method, infer the existence of those gases in our atmosphere. And not only can you infer the existence of those gases, you can also infer the existence of processes that must have put those gases there. So the blueness of the sky tells us we have, a certain amount of atmosphere. It's a certain amount of atmosphere that's scattering blue light everywhere. The extent to which these squiggles go down here on this y-axis tells us how much of that gas is there, how much oxygen and ozone, for example, tells us there might be life on that planet. Methane might tell us there's anaerobic bacteria on the planet. Carbon dioxide might tell us there's volcanic activity on the planet. This is a real spectrum, annotated spectrum of the Earth as it was reflected by the moon and retrieved back by ground-based astronomers. Okay, so we've done this for the Earth. We've looked at Earth as an exoplanet, and we found all the signatures that we can attribute to life and the processes of the planet in addition to life that support its identity as a habitable planet. We can do this also for exoplanets with sufficient instrumentation. And so these are some figures I consulted with National Geographic on looking for life elsewhere and how we would use and infer the light from distant exoplanets to fingerprint the signs of life. So this one is showing you that you can use, this is an infrared spectrum. So this is like the heat spectrum of Earth. And it's showing you that there's methane and ozone and carbon dioxide and water in Earth's atmosphere, which supports the existence of life. If we saw this on a distant exoplanet, we would be very impressed and interested. This is a concept of a planet that is dominated by these rhodopsin-like organisms that are using this alternative pigment in order to capture energy and support their metabolism. And so this is like if the ocean were made very salty and completely inhabited by these organisms. And it's quite a beautiful picture, but that would actually impart a observable signature in the spectrum of the planet that we could detect. And here, really the point is that we can use all those signatures and we can, and observational data and mathematical techniques in order to quantify our certainty or uncertainty about this detection. And this is a little bit complicated. I'm not going to get into this too much, just to say that we can use mathematical techniques for example, say how much oxygen must have been produced on this planet in order for us to see it in the atmosphere. How much methane must have been produced in order to see us in the atmosphere? Is that compatible with an abiotic process or a biological process? And so now I want to get to something that was mentioned by some of our volunteers, including Chris. And it's really important to me, which is using Earth and its evolution as a test case, because Earth represents not just one, but a palette of possible biogeochemical states for rocky exoplanets with secondary atmospheres. And that's because our atmosphere has changed so greatly over geologic time. Geologic time, again, remember that cosmic calendar where we have, the human lifetime is 0.23 seconds. And, only in the half month of December do we have multicellular life. Well, there's a lot more Earth history than that. And so I want to show you this image. On the left is an artist's conception of the Archean Earth. This is Earth 3 billion years ago, before the atmosphere had appreciable amounts of oxygen in it. And it is a methane-rich, hazy orange planet where you have hydrocarbon hazes like we find on Saturn's moon Titan. Because there's no oxygen and so much methane from anaerobic bacteria, you would not be able to breathe on the surface of this planet. So don't go back in time 3 billion years if you ever have a time machine. On the right is Earth today, where we have our blue skies. Organic haze is only from, our industries, and an oxygen-rich atmosphere. And so the point is that on the left here is the amount of oxygen in our atmosphere versus time. On the right is showing the types of biosignatures we might have been able to see on Earth if we were a distant observer, say 3 billion years ago, et cetera. And so 3 billion years ago, perhaps it wasn't oxygen that fingerprinted our planet as a biosphere. It was actually methane in combination with carbon dioxide and a disequilibrium between those two gases that in fact fingerprinted our Earth as a habitable and inhabited planet. And indeed, this has been considered by astronomers who are looking for signatures of life elsewhere. How do we fingerprint the existence of life on planets that may not be exactly like Earth, but certainly could be like Earth in the past? We know Earth in the past existed. We have much more data about Earth through time than we'll ever have for an exoplanet. Okay, so this is really important. And so one of those things that came out of studying Earth through time is how its biosignatures evolved and what combinations of gases and features in the spectrum of the planet would tell you not only is this a habitable planet, but it's inhabited. And so in the Archean, perhaps we're looking at methane and water and carbon dioxide. Today, we're looking for oxygen and ozone and perhaps methane if we can get it, and water. And so the Earth's spectrum has evolved through time, just as the atmosphere has evolved through time. I've also thought a lot, and my colleagues a lot, about how we can tell the difference between a planet that has life and a planet that has an abiotic process that may be masquerading as life. And it's a kind of a complicated thing to do, but we can use models of planetary atmospheres, and we can use models of the development of the solid planet and the biosphere in order to identify the differences between a planet that has, for example, abiotic oxygen from photolysis of CO2, that's a breaking apart of the CO2 molecule, or oxygen from photosynthesis. And looking for those combination of gases is really important. We really want to see things like oxygen, ozone, and methane together. It would be really strong indication of life. And so how are we going to do this practically? So in the coming years and decades, we're going to have more and more capability in order to probe the atmospheres of distant planets. Maybe later this year, perhaps next year, the James Webb Space Telescope will launch, JWST, and it will be able to look at a very small number of transiting planets and search their atmospheres for certain molecules. Namely the TRAPPIST-1 system. So we're going to get a really good look at the TRAPPIST-1 system with James Webb. A little bit beyond that, though, there is a report coming out soon, the 2020 Astronomy Decadal, yes, 2020, delayed due to COVID, that will recommend the next mission that's going to be an ambitious mission in order to find perhaps exoplanet biosignatures. Some of those contenders have the capability, 3 of the 4 have the capability of looking for biosignatures. And 2 of them, I'll mention more deeply, have the ability to directly image planets around other stars. That is, take a little picture and we can take that blue dot, perhaps, and run it through that spectrograph, split its light into component colors and wavelengths, and look for those indications of certain gases that we might infer as biosignatures. So one is called LUVOIR and one is called HABEX. These are very ambitious missions. So LUVOIR, this is a image that shows you the difference between the mirror of LUVOIR, the mirror of JWST, and the mirror of the Hubble Space Telescope. Obviously much more capable. It would actually be able to take an image like this of our solar system from over 30 light years away. And then again, we would be able to run that light through a spectrogram and figure out what's in its atmosphere. There's another concept called HABEX, which deploys this petal-like star shade in space in order to block out the light from the star and reveal the light of the planet, which is outshone by the star by a factor of 10 billion. So it's a very hard problem. It's like looking for a firefly next to a spotlight for miles and miles away. And so this is a modern solar system in true cover like LUVOIR. These instruments are not, we can't do everything with them. We're limited by what their instrumentation is. So we can't look at every region of the wavelength space. We can't look for every signature that there can be, but we can look for some of them. We can look for oxygen, we can look for ozone, water, perhaps CO2 and methane. And the important thing is that with LUVOIR and to a lesser extent with HabEx, we can actually get a statistical representation of the nearby potentially habitable planets. So not only will we be looking at habitable planets, we'll be looking at a wide range of planets, hundreds of planets of different classes. The rocky planets, the potentially habitable planets, depending on the architecture of the telescope, we would be able to get a few dozen to maybe 50 of these potentially habitable planets. And so if we look at 50, we can start to say, maybe a statistical limit on the prevalence of global biospheres like Earth in the universe. Okay, and that is the promise of one of these very ambitious telescopes, is that we can say, okay, let's look for oxygen, let's look for methane, let's look for carbon dioxide in a large sample. And if the large sample tells us that life is very rare, that's a meaningful conclusion. Because it's not dependent on just looking for one thing. We're not just looking at TRAPPIST. We're not just looking at Proxima Centauri. We're looking at this whole population of planets. If you think about even further into the future, there's a concept in an astrophysics report from 2013 called the Exo-Earth Mapper. It'd be like a fleet of LUVOIRs that use a technique called interferometry in order to actually map a planet. So instead of having just that dot, you'd actually be able to see a pixelated version of the planet. That's a way far afield, but it's technically feasible. It's scientifically feasible to do. It would just take a lot of engineering prowess. And so finally, I just want to end with the idea that we've gone through the biosignature, the last parts of the technosignature. I know a lot of people are interested in this. There are people looking for signs of intelligent life. It's a complementary way of looking for life, because while we still don't have the technical ability to image Earth-like planets around other stars, we do have the ability to listen on radio frequencies, or look for optical flashes, look for signs of intelligent life elsewhere that might be able to engineer their way into being more detectable than a planet. And so one organization that's doing that is the Search for Extraterrestrial Intelligence, or SETI. And they are thinking about how do we find signs of intelligent life? And so again, we can look at radio frequencies, we can look for laser pulses if they want to be seen. But I think that it's a very good thing to do, but we can't count on this, because from the cosmic calendar, there are probably far more planets with simple life than with intelligent life. So I wanted to just end by saying, for the first time in human history, we will soon have the capability to detect signs of life across the gulf of interplanetary space. And exciting times are ahead. UCR is part of this. We have some teams of people who are working on different parts of this problem. I mentioned how the Earth's history is a window into the potential states, the potential biosignatures on exoplanets. There's a team here called the Alternative Earth team. It's led by a professor in our department named Tim Lyons. And I'm part of that team as well. And so is other astronomer and planetary science, Stephen Kane, in addition to other faculty members, and looking at the changing atmosphere of the Earth and what that tells us about Earth's biosignatures. So it's a whole team of people from a wide range of disciplines thinking about this question. We're also funded by NASA to think about the seasonal biosignatures, the change in biogenic gases as a function of season, and how that might tell us be a stronger biosignature than those gases alone. So I am part of that effort. I'm also part of an effort to look at planets orbiting M dwarfs. TRAPPIST-1 is an M dwarf. M dwarfs are not sun-like, but redder, dimmer, and smaller. They make up 70% of the nearby stars. This is a diagram that shows all the nearby stars are their sort of relative size and color. The yellow ones are stars like the sun, and you can see far more of them are redder and smaller. And so thinking about this particular effects of, say, the light from that star on the atmosphere of the planet, that's really important because those stars so far outnumber the ones like our sun. And you can imagine perhaps the amount of environments for life around those stars far outnumber the environments for life around sun-like stars. So I think that's good. I'm open for questions.
Great, that was fantastic. Thank you, Eddie. I personally also learned a lot, just from your presentation. I know astrobiology is one of the frontier fields now in astronomy, so we're definitely living in a very exciting time to actually witness a lot of these latest discoveries. So I do have a lot of great questions from the chat. So to start with, from Jeffrey Greenspan, asking how are stellar mass and stellar lifetime factored into the search for biosignatures?
Yeah, that's a great question. So we think about the lifetime of the star. So the brightest stars, if you go outside, most of the stars that you can see in the night sky are actually much brighter and larger and shorter lived than our sun. And it's a bias, they're brighter and so they're easier to see. And so what we kind of think about is there enough time if you're a planet orbiting that star to develop life? And the kind of detectable life that we have on Earth. And so if the lifetime of the star is too short, then perhaps we won't be able to catch a planet with life on it. They're also less numerous. So just from numbers, we're going to favor the stars that the stars that are more Earth or more sun-like or lower mass than the sun, and that's the lifetime part of the question. And you say also the what other property of the star did he ask about?
Lifetime, stellar
lifetime
mass
and mass. Yes, so the mass and the lifetime are intimately linked. To the mass controls the lifetime, the mass controls the brightness, the distance, the Habitable zone from the star, and it controls other more subtle things, like the extent to which the chemistry will break apart molecules in the atmospheres. And so we do study this a lot, and we find, for example, that if you put the Earth around an M dwarf star, for the same amount of biological output of certain gases, you actually get a much higher concentration of those gases. And so if you put the Earth around an M dwarf, you get a lot more methane in the atmosphere. It might be easier to see the disequilibrium between methane and oxygen, for example.
Great, that's a fantastic answer. Thank you. And next question is from Harry Wan, asking, what's the value of just discovering microbes? Intelligent life is what we'd want to compare to answer the classic question of whether we're alone or not.
Yeah, I think, the reason why we want to answer this is because every planet that is going to develop an intelligent being has to go through a phase in which it has microbes. Okay, and so if we really want to contextualize our perhaps lack of finding evidence of intelligent life, then we need to figure out, is that because there aren't many planets with microbes on them? That's an important aspect. I personally find a lot of value in thinking about a planet of microbes. I think the Earth is special. If you think about Earth, at almost any time in its history, did not have intelligent life on it, but it had an intricate web of biological processes, interfaced with planetary processes that was influencing its atmosphere. I think of planets with microbes on them as planets that you could imagine we have no capability of getting to these planets and probably won't in the interminable future, but you could imagine actually living on those planets versus the barren planets. And so I think in order to answer the question of intelligent life, we really need to contextualize, how common is simple life? And then, yeah, I kind of just just think there is a lot of intrinsic value in finding a microbe. I am also interested, of course, in finding intelligent life elsewhere. I think, I could wax philosophical about it, but, I mean, we have, 7 billion people on the planet here that we can talk to, and a lot of differences to work through. And I think, if you extrapolate that to intelligent beings, we have ways to go.
Definitely, I agree with that. So the next one is a slightly technical question. Again, from Jeffrey asking, is nitrous oxide a strong candidate for a biosignature atmosphere gas? And is the simultaneous presence of carbon dioxide and methane in an atmosphere considered as biosignature?
Yes, to both those questions, yes. So nitrous oxide is the result of the denitrification process, the bacteria process. Nitrogen, is taken from the atmosphere. It's fixed. It's made into bioavailable terms of forms of nitrogen. And then, when organisms die or decay, that nitrogen is then returned into N2. But in the step between going from biological, nitrogen into the nitrogen that exists in abundance in our atmosphere, it turns into N2. It turns into N2O, and there's a further enzyme that makes that N2O into N2. But some of it leaks out as N2O, as this nitrous oxide, and it's very hard to get without life. In some very small circumstances on Earth, you can make it abiotically, but in combination with other things, it would be a good biosignature. Bacteria are making it all the time. And then the other question was about CO2 and methane. And yes, so if we think about gas giant planets, they have a lot of hydrogen, and methane is CH4. It's like carbon with 4 hydrogens. And methane is the molecule that those atmospheres want to make in equilibrium, but they don't make a lot of CO2. CO2 comes from volcanic outgassing of terrestrial planets. So Venus's atmosphere is dominated by carbon dioxide. Mars's atmosphere is dominated by carbon dioxide. But besides some claims that really, in some small, some claims of very small amounts of those of methane, methane is not known in Venus and Mars, right? And so the idea is that if you saw both of them at the same time, it would show you that there's carbon in two very different chemical states. And so what you would look for is you'd look for the CO2 and the methane together, and not anything in between, like carbon monoxide. And if you saw that, you'd be very suspicious about what is simultaneously allowing there to be carbon dioxide and methane in the atmosphere.
Great, thank you, Eddie. Next question from Jennifer Chuan, asking, what tools do you use today in order to determine the composition of the atmosphere of planets far away?
Yeah. So right now, the kind of Earth-like planets that we're talking about here, we can use Earth as a test case. We're not really looking. We don't yet quite have the capability of looking at those atmospheres. We can look at atmospheres of gas giant planets, of planets that are kind of like Neptune and Uranus, and look at their composition. And what you do is you do something called a retrieval, and you think about what's the signature you see, and then you have a bunch of models, maybe even thousands of model atmospheres that you run through a computer, and you see what matches that data that you're getting from the telescope the best. What model from your computer matches the best? And those models can be varying complexity. I work with some that use chemistry, photochemistry. And there are very, very different approaches to that. But that's basically in a nutshell. We can use computational models in conjunction with data in order to determine the compositions of those atmospheres.
Awesome. Next question. Well, next questions are actually related. So one question is from Aji asking, is it possible to measure magnetic fields of exoplanets?
Yes, there have been some claims of measuring magnetic fields of exoplanets, and I'm not as familiar with this. It's not my field, but they'd have to be very, very strong in order to measure them. Yes.
Yeah, and this next question is also related from Chris Mahar asking, could magnetic fields of exoplanet also play a part in habitability?
Yeah, that's a good question. This is a very open question on, even on Earth. And we talk about the magnetic field, for example, maybe protecting the atmosphere. On the other hand, you think about Venus has a robust atmosphere, no magnetic field. And so we know that they're related. You know, if you have a planet that's too small, it loses its heat easier, it loses its volcanism, and at the same time, it loses its magnetic field. A planet that has a robust geologic activity and perhaps a lot of convection, like Earth does, and even plate tectonics, then and it spins, you know, and then it has a dynamo and it has a magnetic field. And those things are related, but we're still trying to piece apart, you know, to what extent does magnetic field play in retaining an atmosphere versus even promoting its escape. You know, so the magnetic field actually helps some gases in Earth's atmosphere escape from the Earth. So it's not a clear-cut thing. It's a great question. People are still, you know, thinking about it.
Yeah, those are very cutting edge questions that we're trying to figure out. And since we were already on Venus, there's one question from Gregory Shanos asking, has the discovery of phosphine on the clouds of Venus as being derived from a biologic source been discredited?
It's an open question, you know, and so there, you know, this is the scientific process in motion, you know, so there's a claim and then there are counterclaims and the evidence is evaluated and it takes time. And so I have actually been part of studies that have looked at it and considered that maybe this phosphine isn't there. And that's what our models seem to show. But of course we could be wrong. And so what we're going to do is get more data, get more analysis, and let the scientific process play out.
Yeah, definitely. And two more questions, both from Jeffrey again. One is asking, is it true that ice on asteroids or comets have a different crystalline structure from the ice on Earth?
Yes, it does, and it can, and there are very different ways of making ice. The ice on you know, interstellar objects is of course much different because you don't have the pressure and temperature regime you have on Earth, and also you're constantly being impinged by cosmic rays, and that does affect the structure of the ices on asteroids. There's also different ices at very high pressures, and so the interiors of the icy moons, like Europa and Ganymede, have different forms of ice, and those ices, you know, you may have heard, you know, ice on Earth is less dense than water and it floats, and it's really good for life because otherwise, you know, it would eliminate everything on the bottom. But there are certain phases of ice that exist at high pressures that are more dense than water, and so there are different. You can look this up. There's ice 3 and ice 4 and ice 5 and ice 7, you know, and people play with them in the laboratory.
Yeah, well, that's really cool. Thanks, Eddie. Last question. So I guess this might be actually related to what you were talking about, you know, in the slides. So the question that might be a little bit just out of context. It says, how about the analysis of fluorescence and bioluminescence, maybe in terms of finding biosignature?
Yes, and I this has been proposed by others, and I wrote about it in a book chapter about the potential of using bioluminescence and fluorescence. The only, it certainly does happen that life produces bioluminescence. You can see it, you know, at the beach sometimes with the dinoflagellates. They make bioluminescence. Fireflies make bioluminescence. Corals are fluorescent in UV radiation. The only challenge is how much light is being made as a fraction of the total amount of light from the planet, and it's usually really, really, really small. And so if the whole planet is only 1/10 billionth the light of the of the star planet system, then you have to go from 1/10 billionth to that minuscule fraction of the light from the planet that's from the fluorescence. And so you sort of have to cover the planet with the fluorescent thing that you're looking for in order to be reasonably detectable with these missions that we're talking about, at least in the decade time scale in the future.
Yeah, definitely. Great. Okay. Well, thanks so much, Eddie. That was a great talk, and I think we had a really nice Q&A and discussion, also about some of the future prospects in the field, and I also saw in the live chat, people were very actively discussing different ideas. So that was definitely great. And also I wanted to thank everyone who is still with us right now for joining us this evening. We hope you liked this lecture and learned about something exciting. Let's also give a round of applause to our amazing volunteer who was helping with the chat today. So you will be receiving a feedback survey tomorrow. We are trying to collect, feedback for evaluation, just to know, how we can do better. And you also have a chance to either suggest future topics that you want to learn about through this Cosmic Thursdays lecture series, or you could sign up for our email list to be notified of all of our future events. And you could also, just provide any kind of feedback that you want us to actually read and improve on. So all those feedback will be read by us. And finally, I would like to point you to a couple of sources where you can actually follow us without being on the email list. So let me just quickly share screen. One is the Department of Physics and Astronomy community outreach page. I will share the link in the chat. And here, typically we will have all the scheduled upcoming outreach events here. And our next lecture is actually going to be in October. Again, it's a virtual one and possibly on planetary defense. So that's also a topic that we have never talked about before, and I'm very excited about that. So you could frequently check back this website, or you could simply just go onto Facebook, and we are Astro UCR. So this is an official Facebook page UCR Astronomy has, and every time we will be posting just all upcoming events and also things related to us in here. So just some resources that I would like to share. And finally, I would like to thank everyone again for being with us tonight, and also thank you, Eddie, for a terrific presentation. So we hope to see you again, and let me just quickly post these links in the chat so people could get it, how to follow us right here. Cool. All right. Well, thank you, Eddie. And I think that concludes our lecture today. So thanks everyone for joining us, and we will see you soon, hopefully in October. Great.
Thank you.
Thank you. Have a good night, everyone.